Okay. Right. So today it's our paper 3 part 2 discussion, right? The SEQ paper. Okay. So let's try to do as much as possible in the paper as we can, right? It's a structured essay paper. So as you know, it's important that you stick to exact wordings from the resource, right? The the most important word you have to keep in mind when you're answering structured essay, you have to be precise, right? Strictly the resource, right? Structured essay is strictly the resource because the marking scheme is strict and you have a limited number of words to write, right? So therefore, it's very important you manage your answer nicely, right? You have to manage your answer nicely. Otherwise, you think you wrote, but you haven't, right? You didn't get marks because you thought you wrote, but you didn't, in fact, right? Because you have written something that is not the point, that is the mark is given, right? So I know these papers are a bit difficult than your standard A-level paper. That's the point, right? That's the point. We are with this sort of papers, we are going to explore the resource. We are going to explore the hidden places in the resource that will go untouched, that will go unmemorized or unstudied. So this will sort of clear all those hidden places in the resource that you might end up sort of discovering in the last few weeks before the exam. Then there's no time. That's going to make you panic. And so the main purpose of doing papers is not exactly to understand the theory, but I during my explanations, I sort of almost quickly summarize the theory as well. But the main purpose of doing papers for biology particularly is to sort of see if you have completely sort of explored the syllabus to the level that you can answer any question. That's what you need to sort of do while you're answering these sort of questions. Okay, right. Quickly, let's come to the discussion. All good, right? With audio and video. Right, okay. You might be wondering why I started a bit early today, right? So yes, so this is our proper time, all right? Okay, right. So come to first question. State four characteristics of organisms. These are easy things. How many in total do you have in your syllabus? Right? How many in total do you have? You have seven, right? You have seven characteristics of organisms to choose from. So any four of them would be sufficient. Any four would be sufficient. Okay, right. So anything you can decide. I will write the first four. You can say order and organization. What else? That means, right? From what? From your molecular level to the cell, from the cell to your organism, from the organism to your ecosystem and the biosphere. Um, you have a nice order and organization of things, which is a characteristic of organism. And then, sorry, organisms carry out reactions, right, that can synthesize or break down. That is, there's a sum of reactions that we define as living reactions. That is called metabolism. The organisms undergo growth associated with development. So you have to you cannot leave growth alone. Never you cannot leave growth alone. Growth must be associated with development always. Okay. Then other things you can write anything: irritability and coordination, adaptation, reproduction, right? What else? Yeah, heredity and evolution. All those any four you can write. So 4 marks. Okay. So these are from introduction to biology, right? Based on knowledge in biology for sustainable food production, 3. So actually, there's only 3 given in your resource, right? Today you can go and learn. If you can remember the first day you came to a bio class, maybe you learned about these things. The very first day you came for a biology class, you learned about these things. They can be questioned, not, I mean, yeah, maybe in the level paper as well, right? So sustainable food production, the population is ever increasing, blah blah, and the amount of agriculturable, I mean, the land you can cultivate is decreasing. So what do you have to do? You have to sort of improve everything, no? So one thing you can do is what? Disease resistant, important disease resistant plants and animals. So that's one thing you can do. Then, so disease resistant as well as what at the end of the day, what do you need? You need your harvest, high yielding varieties of plants and animals you can produce. Then, so this is to increase the survivability. This is to increase the yield. And finally, when you have taken the yield, you have to protect it without, I mean, you have to reduce the waste. So post harvest technological methods. Post harvest, you will learn about these in applied biology, right? We haven't done it yet in the revision class. Applied biology, there's a whole section of minimizing food preservation and how to minimize post harvest loss. Are you there? Right? You will learn about that in there. State the main property of water that causes a film of water on the surface to provide habitat for small organisms. Matakadara water skates, right? Water skates floating on, right? They have this sort of ability to stand on water, right? In physics, did you do surface tension yet? No, right? You didn't do surface tension yet, right? So for physics, you will actually do questions. You will see how the surface tension forces are acting on this insect's legs. If you, they are going to give you the sort of their insects' legs as hemispheres, and you might have to do calculations. What is the main property? So it's surface tension. Surface tension is, be very careful. They're asking the main property, right? They're asking main property. Okay. So what do you have to write? The main, out of the main four, what is it coming under? Out of the main four, cohesive behavior. You can send answers, so I can double check myself as well, right? You can send answers to the chat. Okay. Cohesive behavior. Macronutrients that make up 4% of mass, right? So there's 96% is composed of 4: carbon, hydrogen, oxygen, and nitrogen. The remaining, I mean, so actually out of this 4%, there's another section for trace elements, right? There's another section for trace elements, but majority of this 4% is by who? Yes, majority of this 4% is by who? Ken. Calcium, phosphorus, potassium, sulfur, right? Calcium, phosphorus, potassium, sulfur. Okay, next question. The two ends of the phospholipid molecule show different behavior. Explain. What are the two parts of a phospholipid? What are the two parts of a phospholipid molecule? Yes, what are the two parts of a phospholipid molecule? Yes, how do you draw a phospholipid molecule? You have a head and you have two tails. Neither. So, what is the nature of the head? It likes water. Hydrophilic head. Neither. Then there are two tails. These are They don't like water, so they are afraid of water. So you can call them hydrophobic tails, right? Hydrophilic heads and hydrophobic tails. Then what is the composition? What are the sort of, if you take the sub components, what is inside this hydrophilic head? What is inside there? These are things you forget. Huh? These are things you can forget. What is actually inside your hydrophilic head? Mainly, what is there? There's a glycerol here, right? You can't see green, no? Can you see the green color? That green color you can't see, I think. Glycerol, there's a glycerol molecule, right? What else is there? What else is there? Then it's connected to a can. What are the components of the hydrophilic head? There's a phosphate group, and sometimes there can be additional polar molecules such as what? Can. There can be an additional polar molecule such as choline. Yes. Yeah. All these are part of the hydrophilic head. What is the composition of the hydrophobic tail? To be very exact, the hydrophobic tails are the hydrocarbon chains. Hydrocarbon chains of the fatty acids. Because the fatty acids have a carboxylic head here. That head is actually hydrophilic. Carboxylic head is actually hydrophilic. So that is actually part of the hydrophilic head. So the hydrophobic tails are mainly composed of your hydrocarbon chains of the fatty acids. Okay, is that understood? Okay, so moving on to our next question. Name two ends of the phospholipid molecule show different behavior. Explain. So how do you explain this? How do you explain this? You can say hydrocarbon tails are what? Hydrophilic. Then phosphate group and its attachments plus attachments. You can see my handwriting, right? Online people send me only online, right? Attachments are what? Phobic, hydrophobic. I'm sorry. I'm sorry. Yes. Tuck better now. Right. Other way around. Okay. Other way around. Sorry. Hydrocarbon tails are phobic. Yes. And your phosphate groups and its attachments are philic. Yes. Sorry. Okay. Right. Yes. State two structural differences in the two groups of nitrogenous bases bound to nucleic acids. Two structural differences. So they're asking what? By looking at their external structure, what are the differences that you can see? So what are the two groups? What are the two groups that you have? You have your purines and pyrimidines. You can start by saying, you can start by saying what? You can start by saying purines are larger. Ne? Purines are larger. These are smaller. Why purines are larger? They have two rings, or you can say two carbon rings. These have only one ring. Okay. Purines are large. Purines are large. What are the purines? Again, adenine, guanine. What are the pyrimides? Thymine, uh, cytosine, cytosine, and uracil. Yes. Okay. Right. Say the main function of the smallest type of RNA. What is the smallest type of RNA? What is the most complicated type? Okay. What are the three types of RNA, Mulimba? You have T, M, and R. What is the most complicated? What has the most complex structure? Complex structure. You will learn about this in when you're doing molecular biology. If you have already done, and we have already talked about this in our revision class, right? So it has a complex structure that is going to form the ribosome. So the most complex is the rRNA, and mRNA can be very long. tRNA is very small. tRNA is the smallest. Okay, state the main function. Sorry, so they are asking the main function. What is the main function? Your tRNA is like assistant. Imagine your ribosome is like a kitchen where proteins are cooked. Amino acids are put together to form polypeptides to make protein. So its ribosomes are like a kitchen. So the ribosomes have their assistants that are called tRNA. Their job is to bring amino acids to the ribosome, so they can cook polypeptides, right? So they transport amino acids to the site of protein synthesis. To the site of protein synthesis. Transport amino acid to the site of protein synthesis. That is the function of uh, okay, right. You can write ribosomes, right? Transport amino acids to the ribosomes. That's okay, right? I will accept that answer. Okay, but better to stick to um, what is there in the resource book, right? But yes, logically I can accept that. Yes. Name the groups of invertebrates. Okay, we are moving on to question section B. Okay, any questions until now? Any questions you have? Right. What is the shape of the tRNA? It's a three-loop structure, no? Right? Name the groups of invertebrates that appeared during the late Proterozoic. So, yeah, uh, you don't need to remember this, right? According to your new syllabus sort of instructions, you don't need to uh, remember what happened in each eon and era. So, I will give you a free mark for this. Okay. State the era which includes the present time. I mean, yes. So if you actually want the answer, it is the Nidarians. Uh, Phanerozoic, where are we right now? So we are in the Phanerozoic eon. Uh, where are we right now in the eon? What is the era that we are right now? Currently, right now, we are in the Cenozoic, no? We are currently in the Cenozoic era. Okay, we are currently in the Cenozoic era. According to your new sort of instructions in the resource, you don't need to know what exactly happened in each era and neon. So don't think about it too much, but it can be questioned in school papers. Right? It can be questioned in school papers. So Cenozoic era is where we are living right now. Okay. Right. What is the natural taxon? What is the from the domain to species? Sorry, from domain to your species, you have a hierarchy of taxa, right? You have a hierarchy of taxa. What do you mean by a taxa? It's a level in the classification, right? So, can you see your domains in the living world? Your species, sorry, your domains, kingdom, phylum, class, orders. Can you see? No. Those are all some things that we have made up by studying evolutionary relationships. I mean, using molecular knowledge, molecular biological knowledge, and also just by genetical knowledge, we have just come up with these sort of levels of classification. But if you take the living world or the biosphere as a whole, what is the only classification that we can see? Each individual, what can you see? They belong to a species. That's it, right? So we put species into other classes until the level, into taxa until we get to the domain level. So what is the only natural one that is existing? It's the lowest one, species. Okay, got it? Then they're asking what is the phylogenetic species concept? So how many definitions of species do you have in your resource? You have the proper main one, that is the biological species definition. Then you have three additional ones. So according to biological species, which is the most widely accepted, I mean, most you can say makes the most common sense sort of understanding is that organisms who share similar characteristics and has the ability to interbreed and produce viable and fertile offspring, right? So a species should be able to interbreed and the resulting offspring should be fertile. They should be fertile. They should be have able to have their own children. That's what you mean by biological species. So there is morphological, ecological, and phylogenetic. So read about the other two. What do you mean by phylogenetic? Smallest group with a common ancestor. The common ancestor. So actually, all of us are believed to have one common ancestor, right? All of us are believed to have one common ancestor, which is the first, our heterotrophic prokaryotic sort of single cell that was produced in early evolution, right? So that cell is considered as that organism is considered as the first. So we call him Luca. What is it? Right? Luca kila, last universal common ancestor. Last universal common ancestor. That's still sort of a hypothesis, right? මෙහෙම ලූකා කෙනෙක් හිටියද නැද්ද කියන එක. They are still sort of not exactly sure if there was a last uh, sort of last universal ancestor was there, but we believe that, right? There was a common ancestor for all of us. It doesn't matter if you're a tree or a plant or a fungus or an animal or anything, you had a common ancestor that was shared. Okay. Then we come to the five kingdom that was introduced by Whitaker. Robert Whitaker introduced what? The five kingdom classification. After what? What was the cause for Robert H. Whitaker to introduce the five kingdom classification? Anyone? Why did he do it? What was his need? What was the need to introduce five kingdoms? Yes. With electron microscopy, right? Electron microscopy, scientists was able to look into the nucleus level of cells, right? Unicellular cells, even organisms, even they were able to look into the deeper details. So they were able to see that nuclei are not properly organized in some animals, some cells. So they call them prokaryotes, properly inorganized cells that don't have a properly organized cells, they call it prokaryote. Then who had a properly organized nucleus, they call them eukaryotes. So this was the need to introduce this five kingdom classification to put the prokaryotes into a kingdom called what? Monera. He put them into a kingdom called Monera. What was his criteria? What were his criteria that were used by Whitaker? He used cellular organization, as I mentioned here, right? Nuclear organization or cellular organization. He used whether the animal is unicellular or multicellular, right? And also the nutrition, mode of nutrition. Those were the three criteria, right? If you want, we can put numbers in your resource book, right? Follow the resource book while we are discussing and put numbers. What are the criteria used by Whitaker? Criteria used by Whitaker. Okay, right. Moving on. Name a genus with each of the following structures. Okay, name a genus with each of the following structures. Okay. Sorry, yeah, so the word genus has to be here, okay, right? This is the structure and you have to write the genus here, so right, small letter. Basidiospores are found in where? Basidiospores are found in where? It's very easy, no? Right? So they're asking a genus, right? They're asking a genus. So you can come to the conclusion, yes, Basidiomycota, yes, but you have to give an example, right? You can say Agaricus, right? Basidiospores are found in Basidiomycota, Agaricus, puffballs, shell fungi, or any one of uh, those will get you a mark. Conidia. Conidia. Where can you find Conidia? Which fungal phylum where can you find Conidia? Which fungal phylum can you find Conidia? Yes, send me answers. So, usually fungi has two modes of reproduction. They can have a asexual as well as sexual, right? So, if you take phylum Ascomycota, right? Ascomycota, for their asexual reproduction, they produce conidia, right? Can someone tell me, are these conidia? So, conidia are spores, right? We have two types of spores if you take fungi, right? Do you have, what are the two types of spores that are produced by fungi? Based on their sort of location, right? Based on their location, what are the two types that you can classify? You can have spores that are produced outside that are called exogenous, that are sort of produced outside, and spores can be endogenous as well. I think last week or something I sort of showed you the fungal life cycle diagrams and I quickly summarized. Okay. So these conidia are found in phylum Ascomycota. What are the examples for ascomycotes? I can say aspergillus, Saccharomyces, penicillium, right? Those kind of things. Okay, right. What is the main storage food of yeast? Yeast is what? Saccharomyces cerevisiae. Saccharomyces cerevisiae. So, it's a fungus. So, what is the storage food of yeast? They store glycogen, like us. They store glycogen like us. Name a phylum of seedless plants that has the most recent common ancestor with seed plants, and name a plant genus. What do you think? This is a MCQ point. I mean, those sort of theory point that you have to remember, right? And it's a bit counter, sort of logically bit counterintuitive, but you have to study it like that. You're pterophytes, right? Pterophytes. Phylum Pterophyta is more close to your seed plants. Is more close to your seed plants. So, what is a genus that you can? So that means like first part of a full name. They are asking the first part of a full species or a plant name. So this is the generic name. No? So they are asking the genus name. What is the genus that you have in your syllabus for Pterophyta? You have Nephrolepis. Mind you, have to start with a capital. You have to underline. Na? You have to underline. Otherwise, wrong. Understood? Okay, all right. Any questions up till now? All right. Okay. Write the structures of angiosperms corresponding to the structure of cycus given below. So they have given some things of cycus and they are asking what is the structure of angiosperms. So for this structure, what do you have in an angiosperm? Microsporophyll. So specialized leaves, right, that bear sporangia are called sporophylls, right? Phyll කියලා කියන්නේ related to leaves. Sporophyll means they bear sporangia. What is a sporangia? Sporangia are the reproductive structures of sporophytes. So sporophyll, but it's microsporophyll. Microsporophylls, what do they produce? They produce microspores. No, they produce microspores. Usually microspores germinate into what? The male gametophyte. Yes. They germinate into male gametophyte. Male gametophyte produces what? Sperm. Okay. So what do you have instead of your microsporophylls in flowers? What do you have in flowers that is a specialized wall of leaves? What do you have in flowers that is your nicely specialized wall of leaves that is specialized for reproduction in a flowering plant? You have a flower. So in a flower, there's a male part that will produce pollen grains. And inside those pollen grains, what you are going to have? You're going to have the male gametophyte. So instead of your microsporophylls in your flowering plants, what do you have? Yes, you have your male part of the flower. You have your stamens. Stamens include, right, your male sort of parts of the flower, right? Okay, stamen. This is the filament, right? This sort of stylus like thing is the filament, and at the top of it, what do you have? Anther. Anther, right? So these are the filaments, and this is anther, which is the male part of a flower. Okay, right. Female gametophyte. Okay. The thing is, in a flowering plant, you have a female gametophyte. You have a female gametophyte inside the ovule. Inside the mature ovule, you have a female gametophyte. But that female gametophyte is very, very reduced, very, very reduced. And it has how many? What is the composition of the female gametophyte in a flowering plant? 7 cells composed of, sorry, only 7 cells, huh? Right? Only 7 cells and 8 nuclei. So it's a very reduced structure. So we call it what? Yes. What do you call the female gametophyte in a flowering plant? Anyone? Just send to the chat. What do you call it? What did you write? Not sleeping, right? Exam में how many months left? 5. You can send, right? Listening, right? Embryo sac. Embryo sac. Embryo sac. Embryo sac is the name that we give for the female gametophyte. Okay, embryo sac is found within the mature ovule. If you have a mature ovule, there you are going to have the female gametophyte, which is called the embryo sac. In flowering plants only. Okay, in flowering plants, it is called the embryo sac. Okay. Okay, right. Moving on to the next question. Name the kingdom of organisms which is considered as an artificial group containing polyphyletic organisms, right? Kelim, these are MCQ points, right? Direct MCQ points, right? We have sort of put certain organisms that are not nicely matching. For example, your protozoa and algae into one kingdom just for the sake of classification, that is your protists. Right, kingdom protista is your sort of artificial classification, artificial group in the classification. External feature unique to mammals, right? Something that is unique to mammals only, and external feature. So it's a bit debatable. Mammals, what is the first thing that is going to come to your mind? Mammary glands. Okay, so mammary glands, you can say they are internal, right? They are located inside the body, right? So yes, the breast tissue is sort of having outside, but the mammary glands are located inside the breast. So you can say they are a internal structure, but I have given marks for saying mammary glands as well. Some things that are better, you can say sweat glands. Sweat glands is unique, and hair in hair follicles is a unique thing. Other than that, you can say we are the only sort of class with external ears. අපි මේ පිනා කියලා කියන්නේ, right? External ear pinna that you can find only in mammals. You don't need that. So sweat glands and hair, and I have given marks for saying mammary glands as well. Yes, body covered with hair, you can say. Yes. Given, complete the given dichotomous key. Okay, up to that, any questions? No, right? Okay. Dichotomous keys, right? It's very important. I keep trying to make you practice dichotomous keys because something that happened in the 2021 exam, no 2020 exam, there was a dichotomous key, and many students said they couldn't remember how to do a dichotomous key. I know it sounds a bit stupid, but yeah, that can happen. Okay, because you have been studying molecular and the animal form, grade 13 parts, reproduction, endocrine, and things like that. You have forgotten how to do a dichotomous key. Pentaradial symmetry. I mean, okay. So sand dollar, it's an echinodermate. So you can say kelima. That is the only possible one with pentaradial, right? Do not have pentaradial. Do not have. Let's take it to number 2. Do not have. Do not have. Have tentacles. So a nice one could be your hydra because it's a nidarian. So they have tentacles. Hydra. Remember to underline, okay? Hydra, it's a genus name. Do not have tentacles. Let's take it to number 3. Have a worm-like body. You have hookworm, which is a, yes, hookworm is a nematode. Earthworm, it's an annelid. So both of those can be worms who have a worm-like body. So we might have to take it to 4. And do not have a worm-like body. Who is remaining? You have your chiton and frog. So let's take it to 5. Huh? So අපි 4 බලමු. So out of in 4, who do we have? Hookworm and earthworm. Clitellum. Clitellum is a unique feature for annelids. So earthworm do not have a clitellum. We brought hookworm. So you can say hookworm here. Have an external shell. Who has an external shell? Your chiton, which is a mollusk. Right? Or have you seen in the ocean you have this sort of long sort of shells, right? Sort of lying on the beach. Sometimes you can find chitons, chiton shells. Do not have an external shell, frog. Huh? Using general knowledge also you can sort of answer this. Right, moving on to second structured essay question. Any questions do you have? Second structured essay, any questions? Okay, all right. State an important physiological physiological feature of meristematic cells. So there are structural features and physiological features. What are the structural features? They are isodiametric. They have a dense cytoplasm. They have thin cell walls, right? Those are, you can say, they don't have intercellular spaces. You can say those are structural features. If you take the physiology, that means what? Cellular function, no? That's what you mean by physiology. No? What is the physiological feature? Ability to multiply. Better if you have written under suitable conditions. Under suitable conditions. Ability to multiply under suitable conditions. That's how meristematic cells behave. Ability to multiply under suitable conditions. Two other types of plant cells tissues that perform support in addition to collenchyma cells. Supportive function. Who provides support? Yes. They're asking cells or tissues. You can say something like, you can say sclerenchyma, no? Definitely. Sclerenchyma definitely provides a lot of support. What else? Can you tell me a tissue, another tissue? A tissue that is mainly, it has main other functions, but one of their main functions is to provide support as well. Yes. Yes, xylem. Particularly your secondary xylem, if you take. Xylem is a very, very important thing for providing support. If you can remember vascular tissue, one of the adaptations. Xylem tissue, when vascular plants got xylem tissue, it allows them to grow taller. What is the benefit of growing taller? It allows their spores to be dispersed further, right? Proper, so some effective dispersal was facilitated. State two adaptations of a xylem vessel element to transport water and minerals. What is the structure of a xylem vessel element? It is sort of short and wide, no? Like this, bit fat, right? Bit fat, cylindrical, and you have this sort of pits on their end walls, right? Those pits on the end walls are called perforation plates. Perforation plates, right? So those perforation plates are an adaptation to transport water and minerals. So you can say perforation plate. Can you say something else? Any other adaptation of plant, sorry, your xylem vessel element to facilitate the transport of water? Anything else? Any other suggestions? That is not exactly, they say, so they're saying thick lignified secondary cell wall. I mean, that is mainly to give some support, not exactly to facilitate water transport. Neither. What can you say? Yes, they provide support to prevent collapse. Mm, one of the thing is perforation plates because water is able to flow freely through the perforation plates. I can give you marks if you have written. They are shorter and wider, and they have thinner cell walls. Shorter and wider, they have thinner cell walls. That means what? The lumen that is inside is bigger, so that a lot of water can go through them. It's like imagine have you seen those big drainage tubes, right? Drainage, not tubes, drainage pipes. Toilet එකට හිම connect කරලා තියෙන්නේ, right? They have a big lumen inside. No? If they had a very thick wall, that's going to reduce the sort of the diameter of the lumen inside. So they have thin walls to increase the tube that is available for the movement of water. Okay, all right. Next question. Major differences between phloem translocation and upward movement of water. What do you call upward movement of water? That is silent ascent. Xylem ascent. Okay. Xylem ascent. So, what are the differences between your phloem translocation and xylem ascent? Yes. You can say things like this. Your phloem, I always say, your phloem is like squeezing a syringe. You're squeezing a syringe. What's going to happen when you squeeze a syringe? You are going to produce a pressure that's in bio, we call it a positive pressure, right? So, phloem moves by a positive pressure. So, what do we call it? Yes. Phloem translocation is by positive pressure. What else? And what about your xylem ascent? How do you describe xylem ascent? I like to describe xylem ascent like imagine, so it's upward movement of water. So water molecules, I like to imagine them that they are holding hands like this, and the water molecule that is above is pulling the water molecules below, and the one below is pulling the next one up. So it's like holding hands upwards, right? So it's a upward pull. That's something we call in bio. That is something called, you can say, negative pressure. All right, negative pressure. What are the other differences between xylem ascent and phloem translocation that you can think of? Xylem ascent and phloem translocation. I want answers in the chat. So quickly send. What do you think? Yes. You can say xylem ascent moves through a water potential gradient. Therefore, xylem moves from higher water potential to lower water potential. So you can say xylem ascent is according to a water potential gradient. Can say it's a water potential gradient. What do you think about phloem? What do you think about phloem? Yes, what do you think about phloem? You can say it's a pressure gradient. You can say it's a pressure gradient. Sorry. You can say it's a pressure gradient. It moves from higher pressure to lower pressure. Any other answers that you wrote? No. Xylem translocation, you cannot say xylem. It's passive. Not always. Xylem can involve a bit of active transport where xylem can involve a bit of active transport where. Okay, all right. Do you want me to explain xylem and phloem translocation? In xylem translocation, sorry, in xylem ascent, where do you have a bit of active? That what is the place that requires energy in the xylem movement? So xylem translocation begins when you absorb water. Sorry, xylem movement begins when you absorb water into root hairs, right? When you absorb water into root hairs. So water moves passively, yes, but that is because why? From the soil to the root, there is a water potential. Why is the root having a water potential? Because it has a lot of ions, right? Inside the root, you have a lot of ions. So how did these ions get into the root? How did these ions get into the root? By active transport, right? Active transport. Okay. So you can say mineral ions are absorbed actively. So these ions that were absorbed actually contribute to the absorption of water. So that water will be then moved upwards. So you can say xylem, yes, xylem has an active part as well. Done by dead cells is by living cells. Okay, kind of acceptable. Okay, kind of acceptable. What else? What else? Anything else? Shall we move on to the next question and state two requirements to occur guttation? Okay, so to explain guttation, I'll go to. Did I? Yes, I think I explained this last week also. This diagram I showed you last week also, right? What happens in guttation is, what happens in guttation is something like this, right? So you can see, imagine this is the pink pathway. This pink pathway is the movement of water from the soil, from a root hair into the vascular cylinder that is here. Okay, that is the vascular cylinder. Ah, that's good, right? Xylem ascent occurs only upwards. Okay, that's good. Phloem can happen in any direction, right? Phloem can happen in any direction. That only depends on the sink and source. That's good. That's good, right? Xylem has only upwards, and phloem can happen from any sink to any source. Sorry, any source to any sink. Okay. Right. So as I was saying, what happens in guttation is the main reason for guttation is this, Minamaya. What is this? The endodermis that is containing a ring of suberin called the Casparian strip. So when water moves into this vascular cylinder with ions plus water, they are not, they cannot move back into the cortex and then into the soil. Once they are inside the vascular cylinder, they can't come back. Okay. So that's what happens when this water absorption is happening continuously. And at the top of the plant, what should happen? At the top of the plant, what should happen at usually at the leaves? You should have transpiration. Water should be going out, right? Water should be going out at the leaves. So guttation happens because at night the relative humidity is very high. So there is no gradient from leaves, right? From leaves to the atmosphere. There is no water, you can say water potential gradient because the humidity is very high. So transpiration is very low. What's going to happen is in the root, water and ions are getting accumulated, and they can't go back because of the endodermis. They can't go up because they are not being pulled upwards. What's going to happen is they are going to push upwards. It's like a pressure building here. So that pressure is going to push, and they are going to forcibly come out of the what do you call the place that these water and ions can come out? Hydatodes, right? Hydatodes. So what's going to happen is this is my plant form and function tute. So probably what should normally should happen through the guard cells, out of the guard cells, the water vapor should come out. And the other thing is at night, what's the thing? At night, usually your stomata are closed. At night, your stomata are usually closed. So the guard cells are closed. So again, through the stomata, water can't go out. That's why there is going to be a back pressure building, back pressure. That's going to ultimately lead to guttation. So guttation occurs through what? So this is the xylem ascent, right? Xylem ascent begins, right? Xylem ascent begins with the water uptake from the root hair into the xylem. Then these water molecules are holding their hands like this because of the adhesion, hydrogen bonding, and cohesion, right? So they are going to be moved upwards, and they are going to go out of the stomata, and that is called the transpiration pull. The transpiration pull is going to provide the pull for the ascent. Okay. Then phloem, I have explained transpiration not needed. I'm going to, what do I need to explain? Yes, guttation. Can you see? In certain plants, you can see this sort of guttation fluid as droplets on the sort of ends of veins. These are ends of veins. So in this leaf, you can see the veins, right? At the end of the veins, you can see these water droplets. Okay, that is called guttation. So this is not water that is alone coming out. That is called guttation fluid. Guttation fluid can contain minerals as well. But if you take normal transpiration water vapor, that is only water. Transpiration produces only water vapor. Guttation can lead to the loss of minerals as well. Guttation fluid contains minerals as well. Is that understood? Okay, guttation fluid contains minerals as well. Okay. Name two, sorry, say state two requirements for guttation to occur. So first thing, your water is transpiration should be very low. No? Transpiration should not happen, or it can be close to zero. Transpiration can be close to zero. Sorry, should be close to zero. What else? What are the other conditions? Other conditions. You can say relative humidity very high. That is again sort of related to this transpiration being very low. You can say relative humidity being very high. That is another condition that you have to have. Okay. Name two trace elements that cause chlorosis in young leaves. Chlorosis in young leaves. So this happens because the element you can't give it from mature leaves. So there's something called a mobile element, right? If a deficiency symptom you can see in mature leaves, that means the mature leaves sort of sacrifice their nutrients to give them to the young ones, right? But something, if something happens to bad happens to the young leaves, that's because the older leaves couldn't give that. So what are the these sort of elements that can cause chlorosis in young leaves? Yeah, iron can cause chlorosis, manganese can. These are direct theory points, huh? Direct theory points. Okay. Any questions? Right. Part B, state exact location of the pyloric sphincter in the human body. So they're asking a sphincter. Where do you have the pyloric sphincter? What are the two sphincters? Where are the two sphincters located? Have the stomach like this, right? You have the stomach like this. There are two sphincters at the starting of the stomach and at the end of the stomach. So here you have the almost sort of like the heart, no? Heart is sort of here, right? So you can call this cardiac sphincter. Sorry, you can call this cardiac sphincter, right? Then the sphincter that is controlling the right. Okay. Exact location of the pyloric sphincter. So how do you describe the exact location? This is the starting point of what? Your small intestine. That's going to start as the duodenum. Then it's going to go downwards into the jejunum, ileum, blah blah. Right? So this is the duodenum, which is the first part of the small intestine. So you can say at the junction, if you are describing, you can say at the junction between stomach and small intestine. Simple, right? At the junction between stomach and small intestine. Okay, not asleep, right? People. Also, I have to give you a small message while people are here. Please send your answers a bit earlier. Okay, do not send your answers today morning itself because we have to mark. Okay, so please send your answers properly before Wednesday. Okay, before Wednesday you have to send because I send the paper on Sunday usually. And particularly if you are discussing the second paper, that means you had the time to write the paper almost one and a half weeks, right? So you have to send the paper definitely before Wednesday. Okay, otherwise it's going to be difficult to mark. Okay, so you have to be considerate of that. Okay, and the other message is when you are sending answers here onwards in the answer script or the PDF that you are sending, on the top, please write your name, school, and your WhatsApp number, right? Then it's easy for us to manage the papers. Okay. On the paper itself, okay, paper itself, write your name, school, and WhatsApp number. Then it's easy for us to calculate your total marks and other things, and send back the paper, etc. Okay, got it? Right. Write an example for an essential amino acid. Essential means you cannot synthesize these amino acids inside your body, right? Your liver is your main synthesizer of amino acids. Of what amino acids? It can synthesize non-essential amino acids, right? But there are amino acids that the liver cannot synthesize. Those you have to take from outside. In your resource, I think they have given lysine, histidine. Any one of those you can take as answer one. What is an open circulatory system? What is the basic plan of an open circulatory system? What is the basic plan of an open circulatory system? Yes. What do you say? You have a sort of a long heart that has openings with valves called ostia, right? And so there's a blood vessel that is going out of the heart, right? There's a blood vessel that is going out of the heart, and that blood vessel is going to open into chambers where it can, so they transport a fluid. No, what is a transported fluid called? That is this fluid that is being transported, hemolymph. That hemolymph is going to bathe body tissues in this sort of compartment. What is this compartment called? Yes, sinus. It's a sinus that is filled with hemolymph. So after material exchange has occurred between hemolymph and the cells in the sinus, this blood is going to flow back into the heart through these valves called ostia, right? So they don't have blood, they have hemolymph, and their circulatory fluid comes out of the circulatory system. That's why we call it open, right? It comes out of the circulatory system. That's why we call it an open circulatory system. Okay. So how do you describe? There's a fluid called hemolymph which bathes the tissues and organs directly, right? There's a fluid called hemolymph bathes tissues and organs directly. Say the reason for the evolution of respiratory pigments in complex animals. So before you had respiratory pigment. So what is so it's like a package deal like that. Respiratory pigment is like small lorries. Imagine respiratory pigments are like small lorries. Imagine hemoglobin as sort of small lorries. Why do you produce these lorries? Because your highway system or the road system became a lot developed. Why do you develop a bigger road system? Because your country is getting developed, right? Otherwise, you can just have a smaller footpaths, no? Because if your country is not very developed, right? So if your country is getting developed, your road system is getting developed. If your road system is getting developed, you need a lot of vehicles to do the transport requirements. So that's why animals, as they became more complicated, they started to develop circulatory systems. So in those circulatory systems, they needed particular organic molecules that can take oxygen when there is a lot of oxygen and take that oxygen to somewhere there is no oxygen or little oxygen and deliver that. So that's why animals developed circulatory systems, and with the circulatory system, what did you get? You got respiratory uh, pigments. No? So they're asking respiratory pigments. So why exactly did they get respiratory pigments? Yes. You can say, and you can sort of apply a bit of chemistry into this. So, what is the main gas that we are transporting? Yes, what is the main gas that we are transporting? Oxygen is less soluble in water, right? It's less water soluble. That's why we needed special lorries, right? The water can't go in the road by itself, right? පාරේ යන්න බෑ තනියෙන්. You need a special transport lorry or a vehicle. Water is less water soluble, which is so less water soluble. So blood is a mainly water containing liquid. So in order to transport oxygen from respiratory surface, respiratory can be lungs, can be gills, right? Respiratory surface to tissues, complex animals develop evolved respiratory pigments. So if I say it as a complete answer, since oxygen is less soluble in watery medium such as blood, for complex animals, transportation of oxygen from the respiratory surface to the tissues was a problem. That's why they developed respiratory pigments to overcome this problem and to make oxygen transport more efficient. More efficient. Okay. That's why complex animals evolved respiratory pigments. Okay. Right. Next question. You can any connection issue? No, right? You can yes. Any issues you're having? No. Okay. Name the type of histamine secreting cell found in the connective tissue. Histamine is secreted by who? Histamine is an inflammatory chemical that is involved in sort of inflammation, increasing the permeability, so the blood vessels become more leaky. So you can get a edema when you have a lot of histamine, right? It makes blood vessels a bit leaky. Histamine. Histamine is secreted by who? Mast cells. Mast cells. Okay, mast cells found in your connective tissue. Yes, these are direct MCQ points. Okay. What else can you find in connective tissue? What else can you find in connective tissue? If you take a typical connective tissue, if you take a typical connective tissue, what is considered as the typical connective tissue? Quick revision. What is considered as the typical connective tissue? Yes, your areola tissue. What is your typical connective tissue? Your areola tissue or loose connective. Areola or loose connective tissue. What are the cell types? Right? So if you take a connective tissue, you have three main components, no? Right? If you take a connective tissue, you have three main components. No? What are the three main components of a connective tissue? You have cells, right? You have different types of cells. And then what else do you have? You have different types of fibers. You have different types of fibers. And all of this is embedded in what? All of this is embedded in a matrix. All of this is embedded in a matrix. Those are the three components. Okay. So What are the different types of cells that you can find in connective tissue? Yes, cells can. You can have fibroblasts, right? There's a list. You can have fibroblast, then macrophages. Fibroblasts, what do? They secrete the fibers. Macrophages are chelinophagocytic cells. What else? We just talked about mast cells. Then what else do you have? I mean, your macrophages are a type of white blood cell. So you can say leukocytes, leukocytes are found, and also fat. You can have fat cells. So those are the different types of cells, and you can have three different types of fibers: reticular, elastic, and collagen fibers. And your matrix can be a solid, a semi-solid, or a liquid. Even if you take blood, the matrix is a liquid. There's a question. This is regarding the previous one. You want to enhance oxygen transport in large complex animals where simple diffusion is inadequate. So that is more like for an answer for circulatory systems. Why they evolve circulatory systems? They're asking particularly why respiratory pigments. So yes, a circulatory system was to sort of uh, let's say because of the complexity. There was a larger distance, and the most of the cells are not in contact with the external environment. You can even say that, right? But we evolved respiratory pigments mainly because of the less solubility of oxygen. Less solubility of oxygen. Imagine you have a lot of highways. You have a nice circulatory system, but the thing is, you don't have vehicles. So then the highway system becomes useless. So even if you have a good circulatory system, if the oxygen cannot dissolve in it properly and you cannot transport it, then that circulatory system is useless. So that's why we evolved uh, respiratory pigments, which are vehicles. Okay, got it? State the role of histamine in inflammatory response. State the role of histamine in inflammatory response. Yes, it is one of the first things that is secreted in inflammatory response. What is but exactly done by histamine? What is exactly done by histamine? Yes, what can you say? Role of histamine in inflammatory response, you can say two things. It increases the permeability. What do you mean by permeability? The blood vessels become leaky, right? Blood vessels become leaky, increases the permeability, and also the blood vessels are widened, dilated, right? So then, Kian what happens because of permeability? What is the feature of inflammation that is caused due to increased permeability? Right? What can you see from outside? Now, this person is having an inflammatory reaction. Kela, what is the feature that is caused by increased permeability? Yes, can someone tell? Yes. What happens of increased permeability? The blood sort of fluids are leaking out into the tissue, coming out of the blood circulatory system. So you can have a swelling. Edema. Edema කියලා කියනවා. Swelling. Yes. What happens because of the dilation? Blood is coming out, and also not exactly coming out, the blood flow is increased. Blood flow is increased. So because if blood flow is increased through some area, blood is what color? Red color. So there's going to be a redness because of the dilation. Yes. Okay. Hmm. Name the largest and most potent phagocytic cells found in your macrophages. So we are comparing macrophages with who is the other phagocytic cell type that you can find in your body? Your neutrophils. So neutrophils are usually more small. I mean, sorry, they are small, right? Smaller than macrophages, but they are faster. They can get sensitized very fast. They can arrive quicker than macrophages. Macrophages are they are sort of big soldiers. They are like the heavyweights, right? But they are more strong. Okay. Okay. Name the type of lymphocyte. Sorry. Okay, that contributes to internal defenses in innate immunity. Set is main function. Now, be very careful asking about innate immunity, non-specific immunity. What is the only type of lymphocyte that you can find in innate immunity? Non-specific immunity. Yes, you are natural killers. Natural killer cells, NK cells. Natural killer cells. They what do they do? They detect cells, huh? They detect cells with abnormal can surface molecules and kills them. So actually, your natural killer cells are very, very active in killing virus infected cells. So whenever there's a virus infection in a cell, there's going to be abnormal surface molecules in that cell surface. So that will be killed by natural killer cells. Okay. And also, can you tell me another cell type with abnormal surface molecules, another type of cells with abnormal surface molecules? Anyone that should be killed? That should be killed, immediately killed. That is done by cancer, natural killer cells. Okay, cancer cells. Okay, cancer. Cancer cells usually have abnormal surface molecules, and one of the most important defenses against cancer cells that we have are our natural killer cells. To be exact, because of our diet and also the sun exposure and also because of different environmental triggers, we have mutated cells occurring in our body with a large frequency. Every day we begin cancers, right? In our body, cancers begin. I mean, there's a large number of cancer cells originating in our body in a day, but our natural killer cells takes care of them. Okay, so it's very, very important that innate immunity is uh, you can say competent in a person to avoid cancers. Okay, what are complement proteins? Complement proteins are again, part of our innate immunity. What can you say about complement proteins? I want about two sentences. What can you say about complement proteins? They're normally inactive, you can say. So, okay, what happens when they get activated? They are normally inactive, okay. What happens when they are activated? They can cause lysis of infected cells, or you can say invaded cells. They can also cause promote inflammation and phagocytosis. So it's like this. Your complement proteins. So imagine there is a infected cell. There is an infected cell, and there is a complement protein. So your complement proteins are a complex of proteins like this, right? It's like a complex of proteins. So they are usually inactive. When they get activated, they sort of bind to this abnormal sort of invaded cell. They bind here. And what they are going to do is they are going to punch holes. They are going to punch a hole, and through this hole, water can come in. They sort of break uh, break down abnormal cell membranes so that water can rush in. So what's going to happen when water rushes in? This cell is going to swell and going to break, lyse. That is one of the main functions of complement proteins. Got it? One of the main functions of complement proteins. Okay. Right. Two main characteristic features. I know immunity is a bit, you won't be questioned this much in your paper, right? At minimum, sorry, maximum of two sub questions or three sub questions from immunity will be there. But because we are going into a lot of detail, we are doing only grade 12 topics. I included a bit more immune questions. Right? Two main characteristics of adaptive immunity. What are the other names? Specific immunity. You can say acquired immunity. All those are names for your adaptive immunity. What are the things about adaptive immunity you can say? You actually have three. You actually have three main characteristics of adaptive immunity, right? First one you can say it shows specificity for foreign molecules. Again, it remembers and it has specific moves, right? It its responses differ from different type of invasion to invasion, right? So it shows specificity. Right? What do you have to say? It shows specificity specificity for particular foreign molecule. So I'll just write specificity. Then it can recognize. So it has recognition as well, but you can say acquired immunity, sorry, innate immunity also has that recognition of self from non-self. So better, I think you write specificity and memory. That is one of the most important things. Specificity and memory. So he remembers. If I have seen you somewhere earlier, if did you invade earlier, now then I remember what to do to you, right? Okay, that's one of the most important features of acquired immunity or adaptive immunity. Okay. State the function difference in functioning of T cells in autoimmune diseases, type 1 diabetes mellitus, and multiple sclerosis. So those are all actually autoimmune diseases. No, what do you mean by autoimmunity? What do you mean by autoimmunity? It's like our army is going against us. Our army has gone against us, right? So they are destroying our own country's people and useful resources. Okay. So our army has betrayed us. Okay. So that's what happens in autoimmune disease. What is the definition of an autoimmune disease? The immune system becomes active against self molecules of the body and begins to attack the person's own tissues. Okay. Usually females get a lot of autoimmune diseases, right? So many autoimmune diseases are there. So many, right? Almost all glands can be affected. Sorry, all organs. There can be thyroid effects, adrenal effects. So many connective tissue affecting, right? So, what are they asking? Type 1 diabetes, well, what's going to happen? So, they're asking the difference. So, in DM type 1, what's going to happen? Who is the culprit? It's the T cells. T cells attacked who? Insulin producing beta cells. Insulin producing beta cells in the islets of Langerhans. In multiple sclerosis, the culprit is same, the T cells. They attack the myelin sheaths. What's going to happen? Okay, tell me what is the function of having myelin sheaths? What is the benefit of having myelin sheaths? Yes, what is the why do you have myelin sheaths around neurons? Why do you have myelin sheaths around neurons? That is to increase the speed of nerve conduction. No? They increase the speed that a nerve signal goes through the axon of a neuron, right? To increase the speed. So what's going to happen when you destroy that myelin sheath? The conduction speed is going to go down, but actually it's going to impair the functioning of the entire neuron. Okay, because your myelin sheaths have other functions of regulating the neuron, replenishing the neuron, blah blah, right? So the neuron functioning is going to be severely affected. So multiple sclerosis people are going to have motor issues, cognitive effects, so many nervous system related problems they are going to have. Okay. So type 1 T cells attack insulin producing beta cells. Multiple sclerosis T cells attack the myelin sheaths around neurons. Name the enzyme produced by the kidneys to produce, regulate blood pressure. Yes. Yes, there is a form of paralysis. Yes. What is the enzyme produced by kidneys to regulate blood pressure? There's a nice cascade of reactions that is done by the kidneys to what? Regulate the electrolyte balance, finally producing angiotensin 2 and aldosterone also. What is the starting point? Your juxtamedullary cells, sorry, yes, sorry, juxtaglomerular apparatus, I'm sorry, juxtaglomerular cells produce what? Can what is the enzyme? Yes, renin. Renin is the enzyme produced that is going to start this process. Renin. Any questions? Okay, right. 3A. Shall we move on to question number 3? Any questions? Not sleeping, right, people? Yes. Any questions? Let's quickly move on. What is the structure that controls specific physiological, morphological, and behavioral characteristic of organisms? Yes. How they physiological means how they function, morphological means how they appear, and behavioral means how they behave, how they, how they sort of act. Okay. So what is the structure? If you break down into the all the factors that can control these, you sort of find the answer that they are the genes, right? For us, for example, our genome has about 18,000 to 22,000, 18,000 to about 22,000 genes in our DNA. How does the above structure contribute to evolution? What do you think? No, DNA is not a structure, it's a type of molecule. DNA get together to form genes, right? A segment of DNA is called a gene. So you can say DNA, right? DNA, if you just take a piece of DNA, that is senseless, right? It's like a jumble of words, okay? But if you make a word, sorry, it's a jumble of letters, right? But if you make a word with that letters, okay, or sentence, you can say that makes sense, right? So that's what a gene is like. It's a sort of a sensible idea that can produce a function or a feature, okay? Contribute to evolution by undergoing mutations, right? Or you can say undergoing variation. I would give answers for saying mutations. What is the cellular organelle in which the above mentioned structure in 1A is produced? Where do you have? Where do you have your genes? Where you have your DNA? In your nucleus. Simple, right? What is the structure that appears as a diffuse mass in electron micrographs of non-dividing cells? Found in the above organelles. So why are they saying non-dividing cells? Because if you take a dividing cell, what's going to happen? It's going to disappear. There's a diffusely dense stained mass in electron micrographs. Because why? What's going to happen when the cell is about to divide? Right? When the cell is about to divide, it initially is like a diffuse mass. Oh, it's like a mess, like a ball of string. It's like a ball of string. That ball of string is going to nicely organize themselves into what? Nicely organize themselves into a series of chromosomes, right? That is because why? It's because it's ready to be moved. So imagine, I always use this example. Imagine the books that you are studying every day. For example, you're studying for A levels. Are you going to pack your books into the cupboard every day or put your books into bags every day after you have studied? No, because very soon you are going to need them again and again. So, in a non dividing functioning cell, the DNA is always loosely arranged, it should be easily accessible. So, it's like a diffuse mass. But whenever that DNA should be moved, transported, it has to be nicely, neatly, tightly packed. Otherwise, it's very difficult to move. So that's why when the cell is about to divide and you are going to divide the DNA, it's going to be tightly packed. So in a non-dividing cell, you have this easily accessible DNA that is in the form of CAN, chromatin. What are the components in which the above mentioned structure is made of? What is the composition of DNA? Sorry, chromatin. Sorry. What is the composition of the chromatin? Yes, of course, you need to have DNA. And you can say protein, but you can say histones also. Oh, you can say histone or protein, right? State the monomer units of above components. So they're asking the DNA, now, right? If you take DNA, what is the monomer? Deoxy, right? Ribonucleotides, right? So be very careful. Huh? Deoxyribonucleotides. I think I have given marks for saying nucleotides, right? They're asking DNA, right? Deoxyribonucleotides. And if you take protein, you can say amino acids. Okay. Okay. Right. Next question. What is the level of protein structure found in plasma membrane? In plasma membrane, what do you have? Yes, in plasma membrane, what do you have? So there are phospholipids like this. Neil, there are phospholipids. It's a phospholipid bilayer. Here and there, what do you have? Here and there, you can have transport proteins that are sort of having a channel in the middle, right? So if you take that as an example, a transport protein, so it's performing a function. What is the minimum level protein needs to be functional as a transport protein? You can say a tertiary structure. No? That's the level they become functional, unique, three-dimensional structures. Okay. So the tertiary structure is the best. Okay, give another two examples for of protein with above mentioned structure. You can give anything, no? You can have plasma protein, right? Albumin. What else? Structural protein, like sorry. You can have another transport protein such as your serum albumin. What else? A defensive protein, right? Immunoglobulins, bulin, right? A structural protein such as keratin. Anything, any two, you can give. Myoglobin. Okay. State three functions of microtubules. Microtubules. Yes, microtubules are sort of hollow tubes. No, they are hollow tubes composed of, right, 13 columns of tubulin, right? So tubulin protein is arranged like columns, right? Tubulin protein is arranged like columns in order to produce microtubules. Functions of microtubules. What are the functions of microtubules? You know, one thing we just talked about, plays a major role in cell division. No? Plays a major role in cell division. What is the thing done by microtubules in cell division? You can say chromosome movement. Chromosome movement is done by chromosome movement is done by microtubules. What else? You can say they maintain cell shape. They form cilia and flagella. You can say cell motility, right? Those are mobile sort of locomotive structures of the cell. What else? Organelle movement, right? They can shift organelles inside the cell. Organelle movements can be done. Be most precise. Okay, right? Microtubules, there's a list of functions, right? Be more precise. Don't go into uncommon answers. Why do you need to go into uncommon answers? Right? Be more precise. Not precise, but stick to the sort of the most, let's say, yeah, sort of sure answers. Okay, right. Right. Okay. Okay. Following diagram represents how one of the organic molecules undergoes cellular respiration. Okay, undergoes cellular respiration. So this is sort of like a integrated metabolism thing, right? How different substrates can enter different uh, sort of metabolic pathways. Neither. So there is glycolysis here. That is the only hint that you have. So what can enter glycolysis? What can enter glycolysis directly? And that can have two options, right? So there is a product called A. There is, sorry, a product or starting point called A, which you can break down into two. That can enter the glycolysis and downwards. So that is sort of glycolysis downwards. What you're going to have at the end of glycolysis, you are going to have pyruvate. Then you can have the citric acid cycle, things like that. So what is the only substrate that can break down into two? It's always proteins. So proteins can break down into amino acids. Amino acids, they can break down into glycerol. Glycerol, which can directly enter. I'm sorry. Best to take fatty fats. No, best to take fats here. Yes, yes. Best to take fats. I'm sorry. Yes, fats. Sorry, fats. A is fats. So glycerol can enter glycolysis as glyceraldehyde 3-phosphate. Fatty acids can enter the next step. That is what? You can say this is acetyl CoA. So C should be what? This is fatty acids. So acetyl CoA becomes what? What is the end product of acetyl CoA? What is represented by E? E we can take as a process. You can take it as the citric acid side, right? Citric acid side. Okay. So A is fat, B is glycerol, C is fatty acids, D is okay. Um, yeah, better not to take this as citric acid cycle, right? Yeah, let's not take it as a process. Never. Let's not take it as a process because that can be confusing. So let's say fatty acids, but they do no fatty acids actually get converted to acetyl CoA, but by a process called beta oxidation. Okay. So now I'm thinking, yeah, D. If we take C as fatty acids, D have to be acetyl CoA, because fatty acids don't enter the cycle as pyruvate; they enter as acetyl CoA. What do you think? Yeah, D better let's take as acetyl CoA. Okay, I didn't go through it that much in detail. Acetyl CoA විදියට ගමු and D let's take as citric acid cycle. Okay. So what is represented by E? Let's say citric acid cycle. Okay. You can double check at the end of your second lesson. You have this integrated. No, no, you can't take E as oxidative phosphorylation because why? Acetyl coenzyme may goes into citric acid cycle. It doesn't go into oxidative phosphorylation. What do you goes into oxidative phosphorylation? Your NADH and FADH2, right? Acetyl CoA definitely enters into citric acids. Okay, you can't take it as E as oxidative phosphorylation. No. Okay. Right. Next question. What is the compound that B is converted when it enters the stage of glycolysis? What is B? Glycerol, glyceraldehyde, 3-phosphate, G3P. B is converted to glyceraldehyde 3-phosphate before it in the middle of glycolysis. Right? There's an intermediate stage in the glycolysis. Before you get pyruvate, there's glyceraldehyde 3-phosphate, glyceraldehyde 1,3, right? So there's so many sub steps in the glycolysis process. So B is actually entering in the middle of it. Where do you have CO2 release? Where do you have CO2 release? Where do you get a CO2 release? Yes, pyruvate to acetyl CoA. State the stages where CO2 is released. Yes. So you can say E, no? E. Okay, you can say E. Citric acid cycle. You have a release of carbon dioxide, right? So better take that. Okay, right? So tonight itself, take a, I mean, do a small revision of cellular respiration. Okay. Link reaction, okay, but is it given here? Stages. Um, it's not exactly given a number, no? Right? So you can say yes, pyruvate to acetyl CoA conversion. There is a carbon dioxide release. Yeah, okay, okay. Take link reaction as well. Take link reaction as well. Okay, I think I have given marks for link reaction. Okay, take link reaction as well. Okay. Next question. Answer the following questions regarding the given animals. Brittle star, sand dollar, squid, prawn, scorpion. Presence of an endoskeleton. Who has an endoskeleton? Your squid is a mollusk, so no skeleton at all, right? Soft bodied. Prawns and scorpions, those are arthropods, so you can say they have a exoskeleton, chitinous. So your best answers are brittle star and your sand dollars, no? Which are which are what? Which are echinodermics. They are echinodermic, so you can take them. Okay, right. Um, Presence of locomotive appendages. Who has locomotive appendages? I'm sorry, 10. 10 locomotive appendages. I think prawns නේද? Prawns ලාට තියෙනව නේද ගොඩක්? Scorpion is a arachnid. So they have 8 legs. Is it prawns? Yes, prawns. Squids have 8, right? So prawns. Using book lungs as respiratory structure, that's a definitely scorpion, which is a unique feature of your arachnids. Do you know what a book lung is? Do you know what a book lung is? Have you seen what a book lung looks like? This is what a book lung looks like, right? This is a book lung, right? So it's like pages of a book. You have alternating sort of body tissues, and in between you can have sort of cavities with uh, air pockets, right? So there's a page of body tissue, we call them lamellae, and in between those you have air layer and a body layer, air layer, body layer. So it's like a nice complicated structure. So air can enter through here, and there is a uh, hemolymph coming from this side. So there is gas exchange happening because it increases the surface area a lot. So this book lungs is a unique feature of arachnids. You can only find in book lungs only in arachnids. Okay, only in arachnids you can find book lungs. Got it? Any questions? Any other question? Okay. Experiment done to demonstrate the effect of carbon dioxide concentration on the rate of photosynthesis. What is the experiment? Rate of photosynthesis, no? Mm, so you have photometer to calculate rate of transpiration. What do you have to measure rate of photosynthesis? Yes. What is the experiment? So we have respiratory quotient. Yeah. Odor separators, no? Odor separators. So odor separators, how do you use the odor separators? I think I showed you this last week as well, right? Odor separators I showed you last week, right? Not in this tube. Odor separators is not in this tube. Uh, yes. This is photometer. Where is our odor separators? Oh, no, wait, it's not here. No? It's not in this tube. Okay, right. Sorry, it's not in this tube. Anyway, how do you, what is the equipment? How do you use the odor separators? Yes. You usually use an aquatic plant, right? You usually use an aquatic plant. You take a beaker of water, right? And you have a test tube. Okay. Sorry. You take a beaker of water and then you have a test tube. Into this test tube, you are going to put a aquatic plant, right? You are going to put an aquatic plant such as hydrilla or something, and then you are going to connect a capillary tube with a flared end. A capillary tube with a flared end you are going to connect, and you are going to have a sort of a bend like this, then you are going to have a capillary tube, right? This capillary tube is connected to a scale. It is connected to a ruler with the scale, right? And at the end of the capillary tube, you have a syringe connected, which can use to sort of you can use to pull the column of water, right? What's going to happen is with the rate of respiration, there is going to be what carbon dioxide bubbles collected here. Sorry, with photosynthesis. I'm sorry, right? With photosynthesis, there's going to be carbon dioxide bubbles collected here. There's going to be carbon dioxide bubbles getting collected here, and after about t time, you are going to pull this syringe, and you are going to take these bubbles into here, and you are going to measure the length of the bubbles. Then you can measure the rate of photosynthesis. That's what you do. So how can you manipulate this if you want to measure the effect of carbon dioxide? Measure the effect of carbon dioxide on photosynthesis. How do you do it? How do you manipulate this apparatus if you want to measure the effect of carbon dioxide? Yes. So this solution is actually what we call as 0.01% sodium bicarbonate, right? We put sodium bicarbonate, sorry, right? A big mistake here. This is not carbon dioxide here. This is oxygen. Photosynthesis produces oxygen. Okay. We are going to see the effect of carbon dioxide on photosynthesis. Okay. Nobody even corrected. Okay. So the solution that we put this hydrilla is a sodium bicarbonate solution, right? So we can change the concentration of the sodium bicarbonate solution. The sodium bicarbonate solution is going to act as the carbon dioxide source because it's an aquatic plant. So the sodium bicarbonate is going to produce carbon dioxide. That carbon dioxide will be used by the aquatic plant to produce, to perform photosynthesis. So basically, I'm requiring the steps of Odes apparatus. Okay, Odes apparatus steps ටික තමයි ඇත්තටම I require. Right. So I'll just read out. Okay. So what was the question? Demonstrate the effect of carbon dioxide on the rate of photosynthesis. Right. So you can write down, I'll tell you, right? I think we gave free marks, right? Set up the Odus microburette apparatus, right? Set up Odus microburette apparatus. Okay. Then place a table lamp close to the aquatic, second step two, sorry. Make sure that the microburette is completely filled with water. Step two, make sure that the microburette is completely filled with water. Because why? You shouldn't have any air bubbles before you start the experiment, right? You shouldn't have any air bubbles before you start the experiment. It should be completely filled with water. Step three, place a table lamp close to the aquatic plant to provide adequate light. Place a table lamp to provide adequate light. Right? Observe the air bubbles getting observe the oxygen bubbles being released due to photosynthesis and getting collected at the bend of microburette. Okay. Observe the oxygen bubbles released due to photosynthesis and getting collected at the bend of microburette. Measure the volumes of oxygen released by the plant at definite intervals. Measure the volumes of oxygen released by the plant at definite intervals using the syringe. Using the syringe. Change the concentration. Change the concentration of the sodium bicarbonate solution. Change the concentration of sodium bicarbonate solution and repeat the experiment several times. And repeat the experiment several times. Okay. So when you increase the sodium bicarbonate concentration, you keep on increasing until a certain level, you will able to see that the release of oxygen bubbles is going to increase. Because why? Carbon dioxide was the limiting factor, right? Blackman theory, right? So then onwards, it won't increase because maybe the other things will become limiting, such as the sunlight or maybe the chloroplasts are working at their maximum. Okay, so that is how you use it. All right. How does the above factor affect on photosynthesis of a C4 plant? What do you think? How does the above factor affect on the photosynthesis of a C4 plant? There is not much effect. Okay, because why? The C4 plants already have a carbon dioxide concentrating mechanism, that is the Kranz anatomy. So this practical could be a bit difficult, okay, a bit confusing. So above factor effect on photosynthesis of C4 plant, you can't actually observe that much of a difference. So you can say not much effect, something like that, okay, because they already have a carbon dioxide concentrating mechanism. Two assumptions you make when finding the rate of photosynthesis by using the order separators. What are the assumptions, Kianba? What are the assumptions that you make? In these order separators, everything is dependent on what? Order separators, everything is dependent on that the assumption that this oxygen gas that you are collecting in this bin is oxygen gas. And the other thing is, it is the all of oxygen gas that is produced. So, are those assumptions correct? You are assuming all of the oxygen gas is being released. Sorry, oxygen gas released is collected. Neither. All of the oxygen gas released is collected. What is the factor that can affect this? Oxygen gas can dissolve. Right? The gas can dissolve in water. So some amount of oxygen gas will not be coming out because it dissolves in water. That is one assumption. The other assumption is what? Some of the oxygen gas that is produced is being used up in cellular respiration. Right? Some, because the plant also produce the cellular respiration. Some of oxygen is used in cellular respiration of the plant. Neither. Some of the oxygen is used in the cellular respiration of the plant. So those are the assumptions. So we are considering that all of the produced oxygen is getting released and oxygen is not dissolving in water. You assume that all of the oxygen released is getting collected and also oxygen is not being dissolved. Or you can say oxygen is not used up for the cellular respiration of the plant. Oxygen is not being used up for the cellular respiration of the plant. Those are the two assumptions. Right, two precautions you have to take when preparing the setup to determine the rate of photosynthesis. What are the precautions you have to take to prepare the setup? Yes, what are the precautions? You have to make everything watertight, right? Completely fill, right? You have to completely fill without any air bubbles. So, okay, you filled it without any air bubbles. And the other thing, what can happen is what? When you are suctioning out using the syringe through gaps, right? Properly seal the gaps. Otherwise, through the gaps in the tube system, you can get air bubbles and you can have a wrong calculation. Those are the errors that you can have. Okay. So, yes, there is a small chance that even in your paper you can get a practical question, right? So, never take that possibility out. There is a chance. So, since you still have a bit of time, take about few hours and summarize the practicals as well, right? In my revision class, I will do a practical summarization session. Okay, I will let you know when that's going to happen as a special session, practicals. So yeah, I'll stop for the day here. We have another structured essay question and few essays to discuss. I will discuss those essays few in the upcoming discussion session. Okay. So I will have a sort of a elongated discussion session next week. Okay. I will be stopping here. So any questions you have? Any questions? So people who just send their papers, may දැන් යොපු papers ටික I will mark and send. Okay, your finalized marks will also will be sent. And yes, I will send marking schemes to the group. Okay. And your highest marks I will also send to the group. Okay. So please next week onwards send your answer scripts before. Okay. Before Wednesday you have to send. Okay. Okay. Any questions you have? Okay. So we are starting our brand new 2028 batch in the end of February and also very beginning of March. So that means you're officially coming to the very end of your sort of A-level life. You have the second juniors now. Okay. Second juniors will be there in 3 weeks almost. Sorry, in 1 week's time. Okay. In almost 1 week's time, you have the second juniors. Okay. Right. Okay. Then thank you for joining. Any questions? Great. Then let's finish for the day. Okay. Right. Thank you for joining. MCQ4. I'll send discuss this question next week. Okay. Right. Remind me. Okay. Remind me to discuss this question. Okay. Right. Okay. Good night.